Okay, hello everybody. My name is Chris Anderson and I'm a tutor here at Chegg. And today I will be talking to you about active transport. So, to begin, let's talk a bit about why a cell would need an active transport mechanism. So, one thing to remember is that a cell is not 100% cut off from its environment. It needs to be able to bring in molecules or push out molecules in order to perform different kinds of functions, right? And there are a couple of ways that it can go about doing this. So over here, I just have a quick drawing of a single leaflet of a phospholipid bilayer, and you have this molecule that, let's say it's about, I don't know, 400 nanometers. It's extremely small. It's also uncharged. So when you have a molecule of that nature, you're going to have it be able to pass directly through the phospholipid bilayer. Now, the fact that there's two key points to take away from that, and that's that this molecule is extremely small, and really importantly, it is not charged. So remember about the phospholipid bilayer, you're going to have a phosphate head that has charge, and then an uncharged um, fatty acid tail that's going to be very hydrophobic. So because this molecule is extremely small, it will be able to kind of weave its way through or in between some of these phosphate heads. And because it's uncharged, it will be able to pass through the fatty acid portion of the phospholipid bilayer with very little resistance. And with that, it will be able to get through that bilayer and into the intracellular space pretty simply. Now, the ability of a molecule to do that really decreases as that molecule gets bigger or if that molecule has a charge. But the cell still needs these kinds of molecules to be able to get in and out of it in order to function. So, how does it do that? So, evolutionarily, animal cells have developed a way to allow these larger molecules or these charged molecules to enter the cell through basically what's called facilitated diffusion, which is essentially just going to be a protein-mediated form of diffusion that would allow these types of molecules that would be otherwise impermeable to pass the phospholipid bilayer. So facilitated diffusion is going to come in one of two general flavors. It can either be passive or active diffusion. And before we talk about what's different about them, let's just talk about what some of the similarities are. Now, anything under the umbrella of facilitated diffusion is going to have a protein mediator, be it a channel protein, a transporter, or a pump. And with both types of facilitated diffusion, it's going to selectively move molecules in or out of the cell. And when we say selectively move these molecules, it's really just referring to the protein specificity for the specific substrate that it's going to be moving. And that's an important point. Different proteins are going to be able to let different kinds of molecules pass. Like there are different proteins that allow glucose to move from the bloodstream into certain cells. There are things called aquaporins that will allow water to move in and out of the cell. And specifically, what we're going to be talking about today a little bit is how sodium pumps allow sodium molecules, and specifically sodium molecules, to move in or out of the cell. So just a quick recap of some things that you may have heard about passive diffusion, just in really broad strokes, what does that mean? Well, the first thing with passive diffusion is that it does not require any kind of energy input. And the reason for that is simply because it's moving molecules in a way that's already energetically favored. It's allowing molecules to move up. I'm sorry, it's allowing the molecules to move down their concentration gradients. So they're moving from an area of high concentration to an area of low concentration. So that's just normal diffusion. That's just how diffusion works. You don't need to add any extra energy to a system in order for that to happen. It's already spontaneous. One thing to remember about passive diffusion, though, is that it is very fast. Like once those channel proteins open, you can just have a huge and rapid influx or efflux of these molecules in or out of the cell, respectively. And this is one of the key points with, firing action potentials. Like once those potassium channels open, you have a quick depolarization, which allows that, synaptic firing. Well, it's one of the reasons why you have synaptic firing able to move as fast as it does. Another one's going to be myelination, which, well, we'll talk a little bit more about in depth in hopefully a subsequent video. Now, let's talk a little bit about active transport. So, opposite of passive, it does require energy because it's moving molecules in the opposite direction. It's moving things from a low concentration to a high concentration. And we have a quick display of that here. So we have a hypothetical cell that has, let's just say, an internal concentration of sodium ions that's about 12 millimolar versus the extracellular concentration, which is going to be 145 millimolar. And this protein, sorry, this cell has some transmembrane sodium pumps just scattered throughout its phospholipid bilayer. Now, let's just say that this this cell, has a couple of extra molecules of sodium, which we'll just denote by their positive charge adding into that intracellular space. And it needs to move these guys out. Now, like we just mentioned before, the sodium concentration on the inside of the cell in the intracellular space is much lower than the concentration of sodium outside. So this would just mean that it is not energetically favored for these molecules to just spontaneously move to that higher concentration. So how does the cell get around that? It uses ATP. So ATP, just remember, is generally thought of as the energy currency for a cell. It's going to come through and interact with this sodium pump. It's going to be converted from ATP to ADP through the cleaving of a single phosphate group. And by breaking that bond and releasing that phosphate, you're going to release free energy that then this pump can utilize to move these charges, so these ions, out of the cell and up their concentration gradient. So why would the cell want to do that? So like we briefly touched on a little while ago, cells, specifically let's use the example of neurons, need to make an electrical gradient between its intracellular and the extracellular space. This electrical gradient essentially allows the cell to act as a capacitor. So it's allowing the cell to store energy, which it can use for propagating electrical signals and either firing of neurons. Is also, it's how your neurons talk to each other. It's how your neurons can give messages to your skeletal muscles so you can move. It's how your cardiomyocytes, your heart cells, actually communicate with one another, and it's how they synchronize their contractions so your heart pumps blood properly. This is a very important system, and it's actually really interesting how this basically how these potentials are set up and maintained through the use of sodium and potassium pumps, which is something that we won't get into here, but it would be a very interesting topic maybe for a subsequent video, hopefully. And Generally, just the key takeaways from this active transport video is that active transport does require energy. It moves things that would otherwise be impermeable to the plasma membrane through the use of these protein pumps, and it moves them, most importantly, from low to high concentrations. That is a very important aspect of this. So, I hope this video helps to give you a broad overview of what active transport is, and hopefully as well why it's useful. And well, I guess I'll see you guys in my next video. Bye.